Good morning. Alright, I think when I start stream in the mornings, I'm gonna play Wordle. So I want to get better at this. It's honestly embarrassing how bad I am at Wordle. Where's my internet? Hello? Alright. Alright, so I read that slate is a good starting word. Okay, we got an L and an E. Hmm, leads. I'm not opposed to chat helping me. Um, we don't. There's no S, so we'll just start with an L. There's no A. Hmm. 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 I just need to put something down, I think. Um, L-E. I don't even know. Like, what word would it be? E-L. Elope? Is that a word? Right? Yeah. There we go. Not bad. Hmm. E-L. Well, it can't be E-L. There's no E there. So maybe like O-L or L-O. E-L-O. Anyone got any ideas? Hmm. E. Enroll? That's not a word, is it? No, it's two L's. Damn it. I'm so bad at this. Uh, Belier? That's not a word. That's not a thing. John Locke. Yeah. Can I do it though? I mean, I know the E's not there, so that's not a good word. Mm, Loe. I don't know. Someone watching this pack is going to be screaming at, at their screen. Oh, I want to get like a good guess though. I haven't tried you yet. Is that a word? Is that a word? No. That is a word, apparently. Okay. There's no U. Nole. Damn. B-O. Bowel? That's a word, right? Yes. Should we go for it? It's got the O. It's got to be this, I think. Howl. Vowel. Vowel. Jowl. I'm leaning towards vowel. What are we thinking? Is howl even a word? No. Alright. I think it's vowel. Let's go! Is that good? Feels good. Alright. So we are going to pick up where we left off. Mainly, we are focused in on creating the other half of this PowerPoint, which is going to be detailing the results that I've gotten on this enzyme. So that's halm-2. Um, halm-2 is a class 2 lanthipeptide synthetase, which essentially means that it has two um, domains with two different active sites, where one active site catalyzes the dehydrations on serine and threonine residues on a peptide using ATP. That's where the name synthetase comes from. And then the other side is believed to help with the cyclization process for the peptide. Right. But why should we care about this enzyme in the first place? So this is the slide I left off on. So lanthipeptide synthetases, I think you could say LAN-M. No, you probably want to keep it like this. Are the weapons factories for bacteria. The weapons factories bacteria use to inhibit the growth of competing bacteria. So maybe we could do something like all right, so lacto means milk, right? The production of buttermilk and cheese has also become famous as the first genetically modified organism to treat human disease. Alright, I was looking up stuff. Okay. Which makes niacin are found in environments like fermented food. The production of lanthabiotic provides a massive survival advantage in this environment. With new mechanism of action discovered, many bacteria produce substances to gain an advantage over competitors in their highly competitive natural environment. Discovered a new antibiotic active peptide, so-called lanthabiotics. Specifically against their main competitors, the Corynebacteria. Coronae is presumed to mediate a very special mechanism of action. Medical point of view. Yay. All right. We're able to show effects very specifically against bacteria, which are among the main competitors. Specificity is presumably mediated by very special mechanisms of action that we were able to decipher in detail. Penetrates the cell without destroying it, accumulate in the cell, then dissolve. Okay. New mode of action in the bacterial war. Good morning. Good morning, Parakeet. I did my Wordle and I solved it. Alright, so let's look at what niacin's for. Ooh. I think we should probably put this one up. Yeah, it's higher resolution. I forgot. It was like I didn't get to the. It was like right before the last one. So 1, 2, 3, like 5. I'm not. I won't tell you what it is. Um, 34 amino acid residues. As long as it has 57 mer is converted to phenylpeptide. Um, it's so big. And niacin. Damn, did you solve it though? That fast? That's crazy. That took me like 10 minutes to do. Well, most bacteria generally inhibit. Niacin is a rare example of broad spectrum. Effective against many gram-positive, include lactic acid bacteria, spores. Gram-negative bacteria are protected by their outer membrane, but so I need to remember. So gram-positive means it literally just passes the gram test, and it contains like I think gram-positive have like a thicker cell wall. I forgot what I guessed before vowel. You rewind. Gram. I forgot what. I looked this up, but I forgot what the gram stain test. Yeah, I have a thicker layer of peptidoglycan within the cell wall. Gram negative have a thin layer. So is gram positive harder to? No, very easy to treat. Interesting. So the gram positive just has a thick peptidoglycan layer, whereas the gram negative is covered by multiple thin layers of membrane, which inject toxins, harder to kill, quick to develop resistance. I thought you were very intimidating. What does that mean? Like scary? Uh, whatever. I don't like to think of myself as scary, but maybe some people think of me that way. Um, Gram-negative bacteria are protected by their outer membrane, but may become susceptible to niacin action after heat shock. When coupled with a chelator. Okay. Class 1 bacteriocin, very stable at acidic pHs, even more stable at lower pHs. Mode of action, dissipate the pH gradient. Soluble in water. No, it's because they thought you were cool. What? Scary is cool. Since when is scary cool? Since horror movies? Alright, so what do I want to say here? Niacin, LAN, Antibiotic. Not antibiotic. It's lantibiotic. Does it spell that? Yeah. Lanti. It's because they thought you were so cold that it made you intimidated. Oh, I see what you're saying. Yeah, I see what you're saying. Like a girl that's hard to approach. Well, I don't want to be intimidating, honestly. Lanti. That's how you spell it, right? Lanti. Yeah. B-I-O-T-I-C. All right. All right. So what do I want to say here? I will get into the specifics of the mechanism in a bit, but first let me explain why we should care about this enzyme in the first place. Lanthipeptide synthetases are nature's chemical weapon factory for survival. Shown here is one of the most common food preservatives First thing I thought of when I saw lactose was that was a vinyl record. What are you talking about? This? Wait. Oh, you're saying this right here. It looks like a vinyl record. Yeah. That's what you're saying. Yeah, that's true. It looks like one of those. What's the genre? It's kind of like um, I think it'd be like trance, maybe. Like stuff like that. This genre specifically just has like these type of album covers. Yeah, trance is cool. Shown here are the most is one of the most common food preservatives. Um, extends shelf life by suppressing gram-positive spoilage and pathogenic bacteria. Niacin has an E number? What is that? Okay. Where's the food surface from polymer? I wonder if these gram-positive bacteria are developing a resistance to niacin. Yeah, I hate models is really good. They're sick. Or he's sick. He's one of my favorites. So here's one of the most common food preservatives, niacin. Which is produced natively by Lactococcus Lactococcus lactis. Lactococcus lactis. Say that 5 times fast. Yeah, I'm gonna put some on. It's techno and breakcore. Those are like the two genres that help me focus. Yeah, I just don't know what I'd put though. Like, do you have like a playlist or something on YouTube? Yeah, send it to me. Which is produced natively by Lactococcus lactis. Maybe I'll insert an arrow. Hmm. Do I need to have Apple Music to play this though? I was talking about like on YouTube. Like, is there? Because I don't think I can play any of this without it being copyright. Yeah, no, I have a lot of tabs. I need some of these though. I know there's probably some. Uh, I'll deal with it later. It's fine. All right. So I always like to. There's a little bit of color. Maybe like that. So here's one of the most common preservatives, niacin, which is produced natively by Lactococcus lactis. Um, and then I'll probably do like another slide. I don't know, does this really hammer in the point though? Just means that installs like the iron rings upon the peptide and use ATP and DN. So you see here the cyclase domain is blue, dehydrogenase is in red. I'll get into specifics, but let me explain why I showed the enzyme in the first place. Lengthy peptide synthetases are nature's chemical weapon factory for survival. Shown here is one of the most common preservatives, niacin, which is produced natively. Maybe I'll say like it uses. Yeah, no problem. I'm so used to 0 views, it doesn't even face me anymore. Um, so what protein produces niacin? Is kind of what I'm wondering. Such as nice B, nice C, which are modify the precursor niacin to nice T. Okay. So NISB. Yeah, I think I've already looked at this. So what class is this? tRNA dependent lanthobotic. This doesn't really look, well, I mean, alright, let's go to this other one. Bound to non-eliminable substrate analog. That's cool. What, does it make a thioether linkage? That's something. It's kind of interesting, actually. A large class of natural products. Despite extensive research efforts, little is known. Which are often essential for recognition. Class 1 dehydrations are involved in the biosynthesis. So this would be a class 1. And that's just the dehydratase. These enzymes are glutamate from this side chain. Okay. Interesting. This kind of looks like an ATP a little bit. So this attacks there. I wouldn't expect that. So this is the leaving group then. Okay, so what'd they do? So this is quite different from my enzyme that I work with, but shown here one of the most common food preservatives, niacin, which is produced natively like Lactococcus lactis. It uses a class 1 lanthipeptide dehydratase. And I think it also uses, how does cyclization occur? Maybe it's C. Yeah, there you go. Oh, that looks just like my cyclase domain, actually. It's pretty funny. They're just not attached. Hmm. Does it have the leader bound? NISP. What is NISP? Also, where's the leader? Um, what is NISP? Hmm. That might be for the cleavage, maybe like the transporter protein. What? Okay, so it's just NIS, the dehydratase and the cyclase and the transporter. So it's NIST. Okay. Uses two separate enzymes. NIS B and NIS B for dehydration and NIS C for cyclization. Do they have a crystal structure of NIS C bound? Oh my god. Cyclase. Okay. No, it's NIS P. I don't think they have one. Like, what the fuck is NIS P? Yeah, they don't have one, unfortunately. Okay. Which would make this a class 1 lanthanide peptide synthetase, right? I believe so. So class 1, yeah, B and C. So class 2, this one's mine. And then I kind of want to add in, so let's run through this. Halum 2 is what we would call a class 2 lanthanide synthetase. The categorization just means that this enzyme installs lanthanidine rings upon a peptide through the use of ATP and zinc. As you can see here, there's a cyclase dimension in blue and a dehydratase shown in red. I will get into the specifics of the mechanism in a bit, but let me first explain why we should care about this enzyme in the first place. Lanthipeptide synthetases are nature's chemical weapon factory for survival. Shown here is one of the most common food preservatives, niacin, which is produced natively by Lactococcus lactis. It uses two separate enzymes, NISB for dehydration and NISC for cyclization, which would make this a class 1 lanthanidine synthetase. So maybe I'll be like, I'll talk about like the properties of, like I'll make like a list or something. One, promiscuous enzyme, or maybe like substrate tolerant, high tolerance for variable sequence substrates. Another thing is what makes these, I think I asked AlphaFold actually to do that. Or not AlphaFold, but ChatGPT. Yeah. Alright. Dialantipeptides, high structural stability and resistance to proteolysis, antimicrobial compounds, terminal antibiotics, niacin. So maybe we could talk about like niacin and lanthipeptides in general are highly effective against gram-positive bacteria, making them potential how do I word this? Niacin and lantipeptides in general are highly effective against gram-positive bacteria, are shown to be making them promising natural products for exploration. And then I did want to, I think this is cool. There's this paper here. I used GLP-1 GLP-1, which is Ozempic. There we go. Wait. There we go. It's a more recent paper. Using LAN-M enzymes to modify. Okay, so this is mainly talking about. Yeah, so I can mention that after this. Lengthy peptidases are nature's chemical weapon factory for survival. Shown here is one of the most common food preservatives, niacin, which is produced natively by Lactococcus lactis. It uses two separate enzymes, NIS-B for dehydration, NIS-C for cyclization, which would make it a class 1. Niacin and lactic peptides in general are shown to be highly effective against gram-positive bacteria, making them promising natural products for exploration. Regarding the enzyme itself, studies have shown that it is capable of handling peptides with variable sequence length and composition. This makes sense if you consider all right, let's read this for a second. It's often exploited to improve in nature. Natural products belong to a class of robots are known to install a number of highly attractive modifications with high selectivity. That's why enzymes guided to the peptide by corresponding leader peptides. Moved to the last step by synthesis. Here we exploit their leader peptides and their matching enzymes to investigate the installment of D-alanine post-translation. Critical position hormones like glucagon. We offer insight into how the precursor peptide can be modulate. Okay. So they basically took the leader portion, which is important for like substrate recognition for the enzyme, and then substituted the core with GLP-1. That's pretty cool. Able to cleave with other residues. Penultimate position. The alanine is introduced into the core peptide. First, the line recognizes literature contains several other examples using unmodified substrates, but our goal was to alter the specific position and leave no traces behind. All constructs. Yeah. Enzymes found here. This is what I'm going to do. I'm going to copy all of this. Oops. And give it to ChatGPT. Summarize this paper and explain what they found, how they proved what they found, and what this says about LAN-M enzymes and their utility. And bioengineering. All right. All right, class 2 lanthanopeptide synthetases and their role, which are single proteins that carry both dehydrated and cyclation steps needed to mature. Contrast class 1 systems. LAM is a bifunctional synthetase encoded by a single gene. It has 2 functional domains. Class 2 are found in various bacteria and are responsible for biosynthesizing a wide array of LAM peptides. Many of these products are antibacterial peptides, bacteriocins, that the host organism uses to inhibit rivals. Oh, that's cool. Nearly 30 different precursor peptides. Yeah, versatility is one enzyme can recognize and modify numerous distinct substrate peptides to produce a family of structurally related lanthanopeptides. Yeah. Regarding the enzyme cell studies have shown that it is capable of handling peptides with variable sequence length and composition. So maybe we should look at PROC-M then. This is a long paper. When was this published? Oh, super recent. The latter is a cyclic peptide. B is a GLP-1 analog produced by PROC-M. It's resistant to proteolysis by peptidase. Cool. So are these all produced by PROC-M? What is the reference figure 3? Report of the addition. The generation. is also tolerant to hormone peptide sequences, making it suitable in the biomedical field. Okay, so one of these is, yeah, C. Come on. I should probably read this. Arial Nova Light is the best font. Don't argue with me. Get rid of this. High tolerance for variable sequence substrates. I want to put like a picture there. I like the GLP-1 thing. There we go. All right. They show Lin enzymes plus LAGD hydrogenases can be used in E. coli to install a single D-alanine at the defined position in GLP-1, GLP-2, and that you can tune where the enzyme acts by playing with leader core architecture and gly linkers. Here's class 1 and 2, a factory for semi-programmable GLP-1 modifying machine. Penultimate position blocks, mostly preserves potency. Prochem is the only one that robustly modifies at multiple sites. So that's my enzyme right there, Hallam 2. Um, ugh. So maybe let's see if I can find like a good figure. Maybe this one. I need to probably understand what each of these things mean though. I'm going to say this. All right, let's just do the thing and read this paper, I think. Non-cognate cognate substrates, allowing for the inherent flexibility of length peptide enzymes and point character can be utilized to create peptides with improved bioactive and physiochemical properties. Recent advancements in promiscuous enzymes. It's notable given the limited number of characters. Gram-positive bacteria mainly produce lengthy peptides, but bioinformatic. Many biosynthetic gene clusters for lengthy peptides. It is notable given the limited number of characterized lengthy peptides in this group, particularly their antimicrobial activity against multi-resistant drug bacteria. Found positive, possess antiviral, antifungal, diagnostic molecules due to the ability to bind to cell membrane components. That's cool. Food preservative due to strong activity. This knowledge positions lengthy peptides as promising candidates for various clinical and biotechnological applications. Yeah, I should probably take some notes on this. Let's see. Like what is a RIP? Let's just read through this. Precursor peptides divided into two regions, leader and core, such as the production of the peptide by ribosomes. Ribosomes. Whoa. Pretty picture. That's just a GIF. Builds proteins by translating RNA to amino acid chains. Okay. This is followed by the binding of the leader peptides by synthetic enzymes, PTMs. Finally, the protease cleaves the leader peptide, resulting in a mature peptide. 6 distinct classes each characterized by the specific enzymes that facilitate dehydration and cyclization reactions. This concept is a form of enzymatic, where a single enzyme can catalyze the same chemical reaction on different substrates. Both terms are currently used to describe the ability of these enzymes peptides, even if those peptides do not belong to the original cluster. This characteristic is often associated with the leader peptide involved in the process of enzymatic recognition. Additionally, the presence of motifs and secondary structures are crucial for the correct processing of the products. Speaking, I need to start up the next simulation. What? There we go. There we go. In the meantime, we could probably do that. I don't know why it's always so slow. It used to not be like this. Conformation model has been proposed. The leader peptide binds to the bystander enzyme, thereby removing the affinity to core peptide due to low. Okay. Regarding the enzymes, studies have shown that it is capable of handling peptides of variable sequence length and composition. So this is mainly due to the fact that the leader peptide is what does the thing. Okay. All right, let's look at this. There's this one image I'm thinking of. It's by Vander Donk, I think. Oh, does he have a Wikipedia now? Oh, wow. Bigwig. Um, what is this? There we go. That's kind of what I'm thinking about. This is hard to like word. Can I ignore tailoring enzymes to diversify like the peptides? Catalysis without the need for an to function in concert. Require a leader peptide for catalyzing reinformation. Research highlighting the significance of the FXLX motif in the leader peptide. Fewer premature niacin molecules. Crucial leader peptide binding, particularly. Yeah, that makes sense. Additionally, in the system, randomly so assumption that it's capable of enzymes are capable of handling peptides with variable sequence length and composition. This is mainly due to the fact that these enzymes have high binding affinity to the leader portion of the peptide, whereas the core can be more diverse in its sequence. So maybe I can't, maybe I will use this picture here. That I don't have access to. There was this one paper. It was like a thesis, I think. Could just use this. All right, I think maybe here I mentioned it. Yeah, I think it's this one. Maybe this. Damn, I don't know. It was definitely something like this. Why can't I have access to this? I could probably find a thesis somewhere. Maybe this one? Um, Rip biosynthesis. Oh, that's nice. It's not the image I'm looking for specifically, though. Oh, yeah, that's it. There we go. Conformation selection model. Adapted from 157. So maybe I should look at that. I'm not looking forward to writing a thesis. There we go. Ew. Does it look bad? Mm. According to enzyme itself, studies have shown that some specific enzymes are capable of handling peptides of variable sequence length and composition. It's mainly due to the fact that these enzymes have high binding affinity to the leader portion of the peptide, whereas the core can be more diverse in its sequence. This has led us to believe that the enzyme operates through a conformational selection rather than a strict binding mode. So God, how do I do this? it said something in here that like, so with mutagenesis studies, when you modify, like say a residue, it's a lot easier to just change the peptide rather than the um, the enzyme itself, which kind of makes it easier to work on. The evidence is we're showing these enzymes referred to as promiscuous enzymes. It's often associated with the leader peptide involved. Additionally, the presence of motifs is crucial for, although this role varies. Did I change the title? No, it's part 3 again. Wonder if I could just, does that work? Hopefully that's okay. Um, I mean, I could just throw like a bunch of paper titles in here. Could be one option. Therefore, from the modification of cognate, I think it was this. A study conducted in 2016 used to produce 5 new lengthy peptides with high antibacterial activity without need to modify cognate enzymes. Oh. I'm stumped. I don't know. I don't know how to do this. Can't think straight. Binding affinity to the later portion of the peptide, whereas the core can be more diverse in the sequences. In effect, this allowed us to screen allowed to find lantipeptides with high antibacterial activity without modifying the enzyme itself. In effect, this has allowed us to find lantipeptides with high antibacterial activity without modifying the enzyme itself. Reducing the challenges associated with that. Alright, so let's switch gears and about the second half of my proposal, which involves understanding the catalysis behind halidrosine beta synthetase, halin 2. Halin 2 is what we call a class 2 lantipeptide synthetase. This categorization just means the enzyme installs lanthanide rings upon a peptide through the use of ATP and CN. As you can see here, there is a cyclase dimension in blue and a dehydration in red. I will get into the specifics of the mechanism in a bit, but let me first try to explain why we should care about this enzyme in the first place. Lantipeptide synthetases are nature's chemical weapon factory for survival. Shown here is one of the most common food preservatives, niacin, which is produced natively by Lactococcus lactis. It uses two separate enzymes, NISB for dehydration and NISC for cyclization, which would make this a class 1 lantipeptide synthase. Niacin and lantipeptides in general are shown to be highly effective against gram-positive bacteria, making them promising natural products for exploration. From an engineering standpoint, I should probably say, from a the enzyme the system lends itself well to finding more potent as well to stumbling across more potent natural products. For From an engineering standpoint, the system lends itself well to stumbling across more potent natural products. In a variety of contexts. Ranging from antimicrobials to this is mainly due to the fact that these enzymes have a high binding affinity to the leader portion. So let me do this then. I know they got a picture here of that somewhere. They have to. Where are you? Where would it be? Maybe this will have it. Oh, why are you so slow? Could do this. What's so frustrating about this is like I'll find a paper I don't think I need an image from it. And then like 10 minutes later, I realize I need an image from it. And then I can't find the paper because I closed out of it. Because you guys shit on me for having too many tabs. Leader, portion, rip. Oh my god, why is this taking so long? Because I'm downloading something. Something like this. Or this. I like this one. Mm. I mean, I could just make this myself, honestly. So I just do that. Yeah, why not just do that real fast? To like insert shape. Like that. Leader, core. Alright, let's change these colors. Like that. Can I round these edges? Is that a thing? Maybe I need to do that. Yeah, I like that more. Leader peptide's a little bit longer. Maybe like this. Alright, even. There we go. Nope. Sure. Leader core. Then we need an arrow. Should I do a big arrow? Thin it out. like that. Or I could pull something like this off right here. Translational modifications. Mm. Shoot. Alright, let's see if this is done downloading. Okay, that should be. Let's look at a simulation. Where would it be? Homologs. LAN. VMD. Shows cartoons. Is there anything interesting going on? So it's kind of hard to see, but the peptide is this. Let me turn on trajectory smoothing. So the peptide is right up there, if you can see my cursor. So this one's interesting because it doesn't have like it has a non-canonical zinc binding site, which is pretty unusual for enzymes of this sort. Do ATP, DHA, CYS, CYM, ZDB, ZNB. Licorice. Trajectory, 1, 2, 3. So we have our ATP there. We're looking for the cysteine to come close to this DHA, really. If that happens, then we get our PhD. But this is starting from an AlphaFold prediction. So I believe there's a lot of like initial motions that are just trying to restructure these like less confident predictions. Oh. Nah, it's got to go for longer, I think. There's some interesting moves, though. We'll see what happens. It needs to go longer. All right. Anyway, let's finish this. So we'll color this blue. All right, and then we can just directly copy this here. Change this to modified core. Oh wait, sorry, I didn't know you were back. Also just curious, what exactly would be the title of your job, anyways? Yeah. Um, so it would be like computational chemist, basically. Computational biochemist. And I mean, I've probably worked in the slideshow for like, I don't know. I did most of it a while back, and that took like a week probably for like the first half. Really, like the hardest part about this stuff is just like you don't want to put in like unnecessary information, right? Because ultimately, a lot of the people that you talk to don't know, you know, the absolute like specifics of what you work on. So you just don't want to bombard them with like information. It's not, it's hard to know what to put in, really. That's why it just takes so long. Yeah, I'm getting through it. I'm getting through it. It just takes forever, honestly. And I hate doing this stuff. Like I'd rather be like doing research actually, but this is part of it. Um, I don't know. Quite a bit actually. Like here, I'm just kind of covering the background. I haven't even gotten into like the results yet, but I have time though. All right, what color should this be? Purple? That? Yeah, that's fine. I needed a break anyway. Should I make this black? Yeah, I think that looks a little bit better. I just need to just be like that. Oops. Like a light green. Uh, and also there's just little stuff like this that takes forever. Like just like aesthetics. I think it looks better. Alright, I think I'm gonna do a no fill here. Or I could do white. This looks good. What? I want to copy this. Alright, now what does this next say? Proteolysis and export. Pro. That looks good. Um, so the question is, do I want to introduce this here? Oops. Alright, so from an engineering standpoint, the system lends itself well to stumbling across more potent natural products for a variety of contexts. This is mainly due to the fact that the enzymes have a high binding affinity to the leader portion of the peptide, whereas the core can be more diverse in a sequence. Here you see a diagram showing the maturation pathway of the precursor peptide to the RIP, which stands for ribosomally post-translationally modified peptide. I wonder how in-depth I'm gonna have to be. How long do you think this and the questions are gonna take? Hopefully I can get through this entire presentation like around an hour. A lot of these slides are really just for like aesthetics. Like, I'll probably spend, because like some slides you kind of want to like duplicate just to add like a check mark or something, so it's like a bit of like an animation. But you like this, for example. So I'd spend like literally like 1 second on that. So it just seems like a lot of slides, but it's really not. See a diagram showing the maturation pathway of the precursor peptide. Here you see a diagram showing the maturation pathway of the precursor peptide to the REP, which stands for ribosomally synthesized post-translational modified peptide. In effect, this has allowed us to find so because the enzyme conducts mainly uses a leader for substrate binding and recognition. This has allowed us to find lengthy peptides with high antibacterial activity without modifying the enzyme itself, reducing the challenges associated with that. Now I could put in yeah, it does. Now I could put in this paper here. Let's just throw it in. Here is one of many examples I found. Here is a more recent study done on GLP-1. My headphones are about to die. So here's the thing here. So GLP-1 for background is Ozempic. So what we can do is delete that. Turn the enzyme into they turn class 2 lanthanide peptide synthetases into a semi-programmable GLP modifying factory. Alright, is that enough for why we should care? It's mainly due to the fact that these enzymes have a high binding affinity to the later portion of their core, can be more diverse in its sequence. So because the enzyme mainly uses the leader for substrate binding recognition, this allows us to find. Alright. Oops. This is a cool image, maybe. Maybe this. Or should I just leave the title up there? We're just gonna leave it at that. Worry about it later. Okay, so hopefully I have outlined what makes these enzymes interesting. So let's dive into the details of what this enzyme does exactly. First, the peptide will bind its leader portion, and the core will migrate to the dehydroactive sites. ATP then phosphorylates serine/threonine residues in A to whereby subsequent elimination subsequent deprotonation of the C beta carbon, or is that the C alpha? Yeah, I think it's a C alpha. Yeah. Pretty sure. Yeah. C alpha carbon. ATP then phosphorylate serine / threonine residues whereby subsequent deprotonation and the C alpha carbon kicks off the phosphate and leads to a double bond. Sulfur cysteine, sulfur then nucleophilically attacks the double bond with the assistance of zinc, leading to a lanthanethionine ring. Sure. Is that all I want to say there? The mechanism is quite straight. Straightforward, really. But what residues are involved much is still unknown regarding the order in which dehydrations and cyclizations occur, as well as the specific residues responsible for this. We do know some things, however, to help us out. I didn't even delete that. I'm probably going to take a break here, honestly. Get some more tea. So I'll see you guys later.